I want a clear definition for saprophytic fungi, we can say that saprophytic fungi are called a group of fungi that lives in the soil and take the nutrition from the soil. And the main root of these fungi is from the soil, and fungi can grow and propagate on decaying plants in the soil. Conidia means spores. Conidia, or spores of these fungi, through airborne, causing contamination of food, culture medium, plants, animals, and other organisms. And especially clinical specimens in hospitals can be inoculated with these fungi. And also some drugs and etc. Kinds of saprophytic fungi depend on each geographic region are different. So, in all of the world, these group of fungi can live in their soils. In their soil can be lived, but in each geographic area, maybe you have different saprophytic fungi from other sites. For example, in this soil in the Kish Island may be different from the fungi that lives in Tehran or other sites because of the temperature, different temperature, and different kind of soil. This can lead to grow some fungal elements into the soils. And in different seasons, in different geographical sites, and these group of fungi can be different. And so the temperature and climate can play a very important role about the distribution of these fungal elements. So, that is important about saprophytic fungi are these points. Like other microorganisms, this group of fungi has some advantages and some disadvantages. For example, about disadvantages about saprophytic fungi. The first, that is some of the saprophytic fungi can be caused to different and fatal fungal infections among the immunocompromised patients. For example, among the patients with deficiency of T-cell system, or among the patients with tumoral and cause to malignancies, some malignancies in AIDS patients, in HIV positive patients, among the people that had some surgery, deep surgery, and etc. And so one disadvantages about the saprophytic fungi is making some fungal fungal infections among the immunocompromised patients. 2 points is about allergic reactions about these fungal elements. For example, some people have allergy about inhalation of these spores, like aspergillus, penicillium, fusarium, and etc. So, among these patients, you can see some hypersensitivity reactions among these patients that name this state as hay fever. And different, another group of patients can be contact with toxins of these fungal elements. For example, you can use from crops, some crops and foods that inoculated with some toxins of these molds or filamentous fungi. For example, if you eat some crops or foods that are inoculated with some toxins of fungi like mycotoxins, like aflatoxins or fusariotoxins, and etc., you can show some clinical presentation like mycotoxicologic presentation. So the consuming from inoculated crops with toxins of the fungi can be cause to very different clinical presentation among some people. And so these three kinds of disadvantages can be seen among the saprophytic fungi. So in the other hand, you can see some advantages among these saprophytic fungi. For example, some of fungal elements like penicillium, usually penicillium species like penicillium camembert, penicillium roqueforti can be used for making some cheeses. For example, Danish cheeses or Swiss cheeses or blue mold cheeses. So can be used in food industry. And some of penicillium species can use due to some favor for food industrial, like for making biscuits and some cookies, because the favor of the fruit that these penicillium species has it. So, importance of saprophytic fungi and their role in human and animal health is very large, large subjects, both in medicine, in industry, and pharmacology. So although this group of fungi usually does not cause serious infections in people with healthy immune system, usually all of immunocompromised patients are that can be can be caused to these fungal elements, and usually immunocompromised patients maybe show some opportunistic progress in fungal diseases that ultimately can be fatal. Today, this group of fungal infection is very important because preventing of fatal diseases like cancers, different kind of cancers. And usually today some people use from some cytotoxins and some immunosuppressive drugs for treatment of different group of diseases, and especially cancers, and chemotherapy, organ transplantation, AIDS, HIV positive inoculation. These are factors can lead to fungal infections, clinical presentations among the immunocompromised patient. But so today these group of fungi that caused by saprophytic fungi can be very important from the pathogenic fungi, because for pathogenic fungi you have some drugs for treatment of these, but about nosocomial or opportunistic fungi, you cannot some drugs for treatment of these elements. Some species of Aspergillus. Aspergillus is very important saprophytic fungi among all of the saprophytic fungi. Aspergillus is the most saprophytic fungi, especially to different species of this genus, like Aspergillus flavus and Aspergillus parasiticus. Aspergillus flavus and Aspergillus parasiticus are capable of producing toxic metabolites that we name aflatoxin. Do you hear about aflatoxin? Aflatoxin, do you know about aflatoxin? Aflatoxin is the secondary metabolites of Aspergillus flavus and Aspergillus parasiticus that after growth into the crops and food can be lead to producing some aflatoxins that can lead to liver cancer among the people. This fixed.
And this one is medicine. Aflatoxin is the medicine?
No, it's not.
This is from the toxin for
Aflatoxin is a special kind of toxin that produced by some Aspergillus species, like Aspergillus flavus or Aspergillus parasiticus. Usually, with consuming some pistachios and some nuts, because into these nuts produce some toxins. After eating of these, you can cause to mycology, like aflatoxicosis. For example, the Aspergillus flavus and Aspergillus parasiticus are capable of producing toxic metabolites that make these aflatoxin. Aflatoxin are fungal toxin, which is important in development of various diseases. For example, liver cancer. So it is established that these compounds, aflatoxin, can leads to liver cancer among the people, among the human. Some fusarium species, like Aspergillus species, can produce some toxins that we named fusariotoxins. For example, fumonizin. Fumonizin is a kind of toxin that produced by fusarium toxins. For example, the fungus that is the name Fusarium moniliform some countries like South Africa, because in South Africa you have very numerous fields that grow in these corn and cotton seed. So these and also grains. So in these regions, usually fumigation are seen among the crops and can lead to esophageal cancer. So if used from contaminated foods that included some toxins in your diet, you can cause to mycotoxicosis. In addition, some saprophytic fungi have some advantages instead of disadvantages. For example, they are applied in preparation of antimicrobial agent or including antibacterial agent and antifungal agent. For example, penicillin. Penicillin can be synthesized from a species of penicillium that we name Penicillium notatum. That today we name Penicillium chrysogenum. Penicillium chrysogenum is another name for Penicillium notatum that was discovered. And penicillin usually are produced by Penicillium notatum or chrysogenum. And we have another antimicrobial agent like griseofulvin. Griseofulvin is an antifungal drug. Antifungal drug for treatment of dermatophytosis. For dermatophytosis, it is a choice drug for treatment of dermatophytosis. So some antimicrobial agents like antibacterial agents and antifungal agents can be synthesized from these fungal species like penicillin. And also, some of the species of penicillium are used as edible flavors for the preparation of cheese, cookies, and other food, like camembert cheese, Roquefort cheese, or cream cheese. Okay, all of these subjects that I want to tell you about saprophytic fungi is a pre-lab for saprophytic fungi laboratory work or practical work that you have tomorrow afternoon by Ms. Dr. R.D. So you can see some saprophytic fungi under the microscope. I would tell you about 8 cases of saprophytic fungi there, and you can see these fungi under the microscope as a practical work. So before that I tell you about these saprophytic fungi and the method of identification under the microscope to you, I want to tell you about generally filamentous fungi are divided to 2 groups. All of the mold or filamentous fungi divided to 2 groups. As I told you, we have 2 groups for mold fungi. The first group is named hyphomycete. Hyphomycete means filamentous fungi that are seen under the microscope as clearly, without any staining. You can see just by lactophenolcotton blue, you can see all of the fungi as blue color because you use from lactophenolcotton blue, and these fungal agents has no any pigment into the cell wall. So all of these group of fungi is hyphomycetes because of the lack of pigment that the melanin into their cell wall are seen clearly under the microscope. Just due to lactophenolcotton glue, all of these are seen as blue color. So this group of fungi are hyphomycetes fungi. Another group of fungi is named zygomycetes. Zygomycetes because they have not any septum along the filament. It's very simple to identification. Just you see under the microscope, very wide hypha without any septum. So you can identify that this fungus is coinocytic hypha, so it is among the zygomycetes. But hyphomycetes, all of these fungi that are located in this group has septum. So this group of fungi are septated fungi. Hyphomycetes are septated fungi, but zygomycetes are aseptated fungi. And hyphomycetes and zygomycetes are lack of the melanin into the cell wall. So you can see under the microscope without any color or with staining by lactophenolcotton glue as blue color. But we have another group for mold that in the cell wall have some pigment that is named melanin. Melanin in the cell wall can cause that these fungal elements under the microscope are seen as dark fungi, as brown color or black color, because this group of fungi has some melanin in the cell wall. So we named this group of fungi phaeohyphomycetes. Pheo means dark or black. So hyphomycetes included two groups. Hyaline hyphomycetes means without any melanin, and phaeohyphomycetes means with melanin. So hyaline hyphomycetes are not any color under the microscope, just with staining you can see as blue color. But phaeohyphomycetes, because of some melanin into their cell wall, are seen as dark or brown color. Okay? And zygomycetes and hyphomycetes are without any melanin. Okay, phaeohyphomycete means dark hyphomycete, and phaeohyphomycete has another name, mycology fungi. So, if I ask you in the exam, what is the another name for phaeohyphomycete? Response: Mycology fungi. Mycology fungi is dark fungi or phaeohyphomycete. The most important kind of saprophytic fungi that are considered as pathogenic agents and opportunistic pathogens are the first Aspergillus. I want to tell you about 8 agent of saprophytic fungi. The first Aspergillus. SPP means a species. A species. Because Aspergillus has about 900 species. The genus of Aspergillus has more than 900 species. So it is very important saprophytic fungi, and in all of the world are seen in the soil and in air and in water and etc. So Aspergillus spp. These fungi can be isolated from some fruits like citrus fruits, vegetables, especially tomatoes, grains, pistachios, and etc. So in the refrigerator, usually you should see some species of Aspergillus on the surface of some fruits like oranges, like tomatoes, and etc. So it's the most common fungus that usually are seen in the environment. The species of these fungus are readily available from the surface of fruits. Conidia in this fungus, which have antigenic properties, conidia or spore of Aspergillus, has some antigenic properties that can cause hypersensitivity reactions, like hay fever. As I told you, in some people with atopic statement, these individuals can cause symptoms of hay fever. For example, sneezing, runny nose, cough, and etc. So some of people show some hypersensitivity reactions after inhalation of these spores as hay fever. Growth of Aspergillus fungi varies according to species, but are often fast growth. Generally, all of the hyaline hyphomycetes has fast growth. About 2 up to 3 days they can grow into the culture medium. But dart fungi usually has very, very slowly grows, about 10 up to 14 days. And the temperature of the growth in all of the saprophytic fungi is about 25 degrees centigrade, like a room temperature. Colonies are cultured in medium for 2 or 3 days because they are very fast. You can see in this picture some 4 different types of species of Aspergillus. For example, this color show you that this species is named Aspergillus flavus, usually inoculated some pistachios and can produce aflatoxin into this. These colors show you that the species is fumigatus, Aspergillus fumigatus, can usually lead to invasive aspergillosis in lungs, about their immunocompromised patients that are caused to malignancies, some malignancies, usually blood malignancies. These colors show you can be related to the Aspergillus treus. For example, different colors about some different colonies can help you that you identify the species of Aspergillus before examine under the microscope. Okay. The term of Aspergillus in Latin language means sprinkler. So, because of the shape of the Aspergillus, the head of Aspergillus, as you see, is very similar to a sprinkler. So in Latin, this name is called about Aspergillus, means sprinkler. I tell you about identification keys, about identification of saprophytic fungi that you can identify these fungal elements under the microscope. For example, if you have Aspergillus under the microscope, you should see the first one, conidiophore. This is conidiophore, means a base that these protein body, these structure, reproductive structure, are located in the head of fungus. So this is conidiophore. This is this raised part is named vesicle, and in top of vesicle you have one row or two rows of parts that you can name strigma or phyali. Strigma or phyali. So you have conidiophore. In top of the conidiophore you have vesicle. In vesicle you have one row or two row according to the special species that we name strigma or phyali, means this base form. And in top of the phyali you have several chains of conidia. So conidia that are located and attached to the phyali or strigma, and they are located on the top of the vesicle, on the surface of the vesicle are attached to the conidia. So this shape can show you that this fungus is aspergillus. As you see, this is very similar to sprinkler. Aspergillus can cause some different fungal infections, like this one, involvement of the nails by some saprophytic fungi. No dermatophytosis of the nails. It is differ from dermatophytosis of the nail because the causative agent of this form is named aspergillosis, and by is done through some aspergillus species. So, because generally the term of onychomycosis means involvement of the nails by fungal elements, all of the fungal elements, dermatophytic agents, saprophytic agents, and yeast. All of the fungal elements that cause involvement of the nails, toenails, and fingernails are named as onychomycosis. Onycho means nails. Mycosis means fungal infection. But their dermatophytosis is a kind of onychomycosis that are caused by dermatophytes. And this form of fungal infections is caused by saprophytic agents like aspergillus. And all of these figures are in onychomycosis, generally onychomycosis. Okay? So you can see involvement of the toenails, and also aspergillus can invade to corneum of the eye, and can cause to blindness among the farmers usually. If contact with dust or some branches of the trees into the eye of the farmers can cause to keratomycosis, means that will show you involvement of the corneum of the eyes, as you see. Usually, this form of fungal infection caused by Fusarium solanum. Fusarium species usually can cause keratomycosis. And under the microscope, usually the nail specimen, you can see this figure because it is the mold, so you can see some hypha under the microscope. Hypha with septate, with septum. So aspergillus under the microscope usually are seen as septated hyphae and can cause these fungal infections. ببخشید چی دارید توی این گوشیتون
سرویس اینجا
چند سویس کردم
دو تا دانه
خب اشکالی نداره دیگه 4
آره دار
بسیار. خب Another saprophytic fungi is penicillium. In penicillium species, like Aspergillus, you can see different fungal infections that caused by these fungus. Penicillium in turn, in Latin language means broom, or and very similar to this view of a microscopic view under the microscope is very similar to broom or fork, because the shape of this fungus is very similar as fork or broom. So the name of penicillium means broom. Under the microscope, usually you can see the a picture like fork or broom. As you see, you can see this figure for penicillium. This is conidiophore. This is conidiophore. In the top of the conidiophore, you can see phyllide, 3 branches phyllide, and attached to the phyllide, you can see some chain of conidia. So this is very similar to broom and fork. Okay. The colony of the penicillium, as you see in the top of the cheese, this is these shape of colony, blue-green colony color on the surface of the colony, and in the center of the colony you can see some eggs. چیزی نریزم رو سر همیشه اینجوریه
از دیروز نمیدونم
انتم مؤتمر
الان
As you see, the broom or fork is the name that belongs to this fungus. Another saprophytic fungi that we have in the practical world is Scopulariopsis. Scopulariopsis. Scopulariopsis in Latin language means broom, like penicillin. Means, as you see in the next slide, show you that the shape of this fungus is the same as penicillium, and it has 3 branches. So the colony, the color of the colony of Scopulariopsis is creamy, tend to cinnamon color and powdery. Powdery form of colony shows you that it is a mold. Mold usually show you the texture of the powdery. And this form, as schematic form, show you that 3 branches is very similar to penicillium, but in big size. If you see one conidia of this fungus, if you see just one conidia under the microscope, you should identify this is a Scopulariopsis. Why? Because this is very large conidia, lemon-shaped and spiky. Lemon-shaped and very big size. So if you see just one conidia of this fungus under the microscope, you should identify that this fungus is Scopulariopsis. And as 3 branches form, it's very similar to penicillium and very similar to broom. But usually under the microscope, slides that show you tomorrow, usually we have one branches that are not similar to broom. But as you see in this picture, you can see a chain of macroconidia that each of conidia usually show you that it can be related to the scopulariopsis because it has two valves and very big size and spiky. It is spike. Okay. Fusarium. Fusarium is another saprophytic fungi. Fusarium means show you that the conidia is very similar to banana or fusiform. Fusiform or banana form conidia is related to the fusarium. And the colony of this fungus usually is white color, has white color, gradually tend to the pink or purple color. And the back of the colony is purple color. This fungus has two different conidia, microconidia and macroconidia. Microconidia is not important for identification, but macroconidia is important. Macroconidia about the fusarium is fusiform. Fusiform or banana form and multicellular, as you see in this picture. If you see one macroconidia of this fungus, you can identify that this fungus is fusarium because it has a fusiform. And in this picture, completely is clear that you can see multicellular cell, macroconidia. It's a very large conidia that usually is multicellular. In this picture and in that picture, show you keratomycosis, involvement of the corneum that can cause to keratomycosis or fungal keratitis with causative agent of fusarium. In more than 90% of cases, the causative agent of keratomycosis is fusarium species, especially fusarium solani. Fusarium solani is the most common causative agent for keratomycosis. Mucor and rhizopus. Mucor and rhizopus are belong to zygomycosis, zygomyceta or zygomycota. Okay, mucor and rhizopus that I told you before. Mucor and rhizopus are from a group of zygomycota, and this group has not any septum across the phylum. And these fungi are very similar together. Just by a method you can diagnose these fungi from together. Mucor and rhizopus can make a bag or sac that is named sporangium that are included full of sporangiospore. More than 10,000, 100,000 of sporangiospore are included into the sporangium. So in mucor and rhizopus, you can see one sporangium, including more than thousands and thousands of sporangiospore, and these sporangium are located and attached to the sporangiophore. چه خبر؟
جنگ شد خبری
نه
نفر دیگه اسرائیل
نمایشگاه میگن که چیز نمایشگاه نیست، یه چیز دیگه بهشون میگن. مثل مانور مثلا یه جور نمایشی یه جور قدرت نمایی. مانور. Okay. Let me follow. So, Mukor and rhizopus. Both of them has a sporangium along with a sporangiospore that are attached to the base that is named sporangium. And in the culture menu, usually you can see this texture, woolly form, and it has very quick growth, and less than 18 hours or up to 24 hours can be feed all of the culture media. So, Mukor and rhizopus can lead to systemic fungal infections that we named mucormycosis or zygomycosis that usually you should see in COVID-19 patients. If you remember, in COVID-19 patients, always some of patients can cause to mucormycosis or zygomycosis because these group of fungi are immunocompromised patients. And in culture menu, you can see this view, this macroscopic view, and full of woolly texture. As a dark, the first white woolly as a color, white color to tend to gray color. You can see a peach that is inoculated with Mukor. As I told you, in this fungus, you have sporangiophore, sporangium, and sporangiospore. And between Mukor and rhizopus, there is one difference. In Mukor, just you see under the microscope, just a sporangium that are attached to the sporangiophore, and into the sporangium are included full of sporangiospore. But in rhizopus, in addition to this view, you can see some rhizoid in the end of a sporangiophore, like this one. As you see in this picture, just you see a sporangium, a sporangiospore, and a sporangiophore. Okay? Do you see any rhizoid in this picture? Rhizoid like similar to root in the plants? Just you can see a sporangium, a sporangiospore, and a sporangiophore. But in rhizopus, with this microscopic view, woolly texture, and gray tend to yellow in rhizopus. And in microscopic view, you can see some rhizoid in the end of a sporangiophore. Okay? So, Mukor and rhizopus. In Mukor, if you see some rhizoid in the end of the sporangiophore, you should name this fungus as rhizopus. Rhizopus has rhizoid. But Mukor has not any rhizoid in the end. Okay? So, this is identification keys for identification of Mukor and rhizopus. Rhizopus, you can see some rhizoid in the end of the sporangiophore, but in Mukor you cannot see any rhizoid in microscopic view. As I told you, among the COVID-19 patients or diabetic patients, as you see, mucormycosis or zygomycosis can lead to very fatal and dangerous fungal infections. As you see, after inhalation, the spores can go the up, in the top of the nose, and transfer to the eye cavity, and can lead to the blindness. And after that, after go to the brain, can be caused to infarctus, and patient can be go to coma. And after 1 week, patient can be died. So we can say that the most dangerous and the most quick fungal infections among the fungal, all of the fungal infections is mucormycosis or zygomycosis. It's very fatal among the, especially diabetic patients, or among the blood malignancy patients. Okay, we have 2 dark fungi. The first, Alternaria, and after that, Cladosporium. You have in the practical world 2 dark fungi, Alternaria and Cladosporium. The first, Alternaria. Alternaria, how we can diagnose Alternaria under the microscope? Macroponidia is multicellular and very like to the blackberry. Because of the presence of the melanin into their cell wall, you can see the color of brown on the macroponidia. So, because this group of fungi has some melanin into their cell wall, so all of these fungi, these dark fungi, has black colony, because show you that this group of fungi has melanin in the cell wall. And the first, the color of the colony is olive color, and gradually tend to the brown and dark and black color. And back of the colony is black. So, show you, this color show you that this fungus is among the dark fungi, means Dimatiaceous fungi. Under the microscope, you can see the shape of blackberry under the microscope, like this figure. These are macroponidia that attached as a chain, and this chain is attached to the mycelium, one hypha. Okay, you have one hypha that are septated and end up in this hypha. You can, the chain of the macroponidia that attached together can be separated from together. And if you see one macroponidia under the microscope, because of the color that is brown and multicellular form, you can say that this fungus is Alternaria. It's very similar to blackberry. Okay. Cladosporium. In Cladosporium, that is another dark fungi. This fungus is very, it has a very slowly growth into the culture medium, and because of the melanin into the cell wall, also this fungi, this fungus is seen as dark colony, and the first it is olive color. It has olive color and gradually change into the brown or black color. As you see in this picture, if you see under the microscope the view of a branching tree, that the conidia is very similar to leaf of the tree. Okay, conidia in Cladosporium is very similar to leaf of tree, and this fungus is very similar to branching tree, like this figure. Branching tree, and as you see, Conidia is very similar to leaf form. So, because of the melanin, the color of this Conidia is dark. Okay, the last saprophytic fungi is Rhodochrona, the last, and after that finish. Rhodochrona is a yeast, just single fungus among the saprophytic fungus is yeast and Rhodochrona, and especially the species of Rhodochrona means Rhodochrona rubra, usually is seen into the microbial culture medium as orange color. If you see, if you have microbiology and you see some bacterial culture medium, occasionally you see some colonies that are shiny and mucoidal for like bacteria that is orange, that has orange color. It is named Rhodochrona, especially Rhodochrona rubra. Rubra means red, and because the color of this fungus is pinky orange, that is named salmon color, so it is taken from the Rhodochrona rubra. As you see, this plate show you the color of this fungus. That the yeast show you that is it has mucoidal colony, mucoidal creamy colony like bacteria, and due to the color, its color that is salmon color, so we named them Rhodochrona rubra. Under the microscope, you can see this view. As you see, it is yeast, and it yeast the body that has one capsule around it. It is a yeast with capsule, capsulated with body, and it is shown as this form. So the positive agent of some infectious disease, like rhodothrolosis, this positive agent is named Rhodochrona rubra. That is under the microscope, you can see this form of fungus. So it is just single yeast form, saprophytic fungi. Okay, it's finished. If you have some question, please ask.
Thank you.